All right, guys. So yesterday we were talking about photosynthesis, and one thing that I wanted to review because we were dealing with energy was trophic levels and energy levels and stuff like that. Most of today is going to be on cell respiration, but I want to look at what's the word trophic mean? Like heterotroph, autotroph, trophic level. They all have to do with energy or food, okay? Eating or food. So if we're talking about a food chain or a food web or a trophic pyramid, we know it looks something like that. And down at the bottom we have the primary producers. If something is a producer, that means it's what? It's doing photosynthesis or maybe what else? Chemosynthesis. But it's taking energy from some outside source and it's using it to make some kind of organic food, sugar usually. And we know the things that can be producers are the plankton, the algae, some bacteria, and plants. The things that eat those, here they call them herbivorous consumers. What have we been calling those things? Primary consumers. And then these would be secondary, tertiary, quaternary, and top level, or we could go on and on. But the thing we know about each level in this pyramid is it gets smaller as you move up. And the reason it gets smaller as you move up is it's mostly about what? The amount of energy available decreases as we go up. The things that eat the plants use some of the energy from the plants. But most of that energy that the primary consumers take in from the primary producers, most of it gets lost as heat during what? During cell respiration. So that it's not ever passed on up. Heat loss every time down. What percentage of the energy that started in the bottom level makes it to the second level? Only 10%. 90% is lost. 10% is transmitted. That's the rule of 10. So by the time you get up to the top, there's not a whole lot of energy left. What do we call, what's another word for heat that's released into the environment? Entropy, right? It increases entropy. But that's a trophic pyramid. And know that whole rule of 10 thing, that only about 10% of the energy in one level is transmitted to the next. And here you can kind of see it broken down in numbers. What's missing here that probably ought to be here? Yeah, there's no producer level in that pyramid, right? That freshwater shrimp are definitely not producers. Probably ought to be another level we're just not seeing down there. All right, let's pull up some respiration stuff. All right, question. What organisms do respiration? Every living thing. Now, we're looking at up here an equation for aerobic respiration. Does every living thing do aerobic respiration? No, most do, but few don't. Few things like some bacteria, some yeast, they don't necessarily do aerobic this way. Do we do aerobic and anaerobic, or only aerobic, or what? We do both. Here's an equation, like I said before, for aerobic respiration. Make sure you know it. You've got to have some kind of food source. Is that food source always necessarily glucose? No. Turns out you can start with other things, and we'll talk about how that happens in a minute. But usually we start with some kind of sugar. We have to take in oxygen. We need some ADP and some P, so we can make ATP. And the waste products we give off are carbon dioxide and water. But the purpose of cell respiration is to make that ATP. Does cell respiration make energy? Nothing can make energy, right? It takes energy from sugar and transfers it to something that our body can actually use, which is ATP. It's about moving energy, not about making energy. Now you guys should know already the steps of respiration. Before we get into that though, Scientists think that the first things on Earth were bacteria, right? And then the bacteria supposedly became the eukaryotes slowly because of what? Endosymbiotic deal, right? Somewhere along the way, some of those bacteria evolved the ability to what? Do photosynthesis. And that released what out into the atmosphere? Oxygen, which they don't think was there before. And once oxygen was out there, some things evolved to use that oxygen. And those things would have been the first what? Not necessarily. They're aerobes. First things to use oxygen. First things that did aerobic respiration. One other thing to mention, cell respiration works basically the same way in all organisms. It's one of those what? Yeah, one of those characteristics that's conserved through generation after generation after generation, species after species after species. A good example of common ancestry, right? Let's talk about aerobic respiration first, and then we'll work our way down, maybe to anaerobic today. I don't know if we'll get there or not. The first step in any kind of respiration, aerobic or anaerobic, is glycolysis. And you look at that name, glycolysis is first of all, what does the word mean? Split sugar, split glucose, break it in half. Whenever you break a bond, typically what is released? Some energy. But it's not just the process of splitting. What else are we doing to glucose? Yeah, by how do we usually take the energy out of things? Oxidation. Take away electrons. So both of those things are happening. We're breaking some bonds, and we're doing some oxidation. Glycolysis happens in the cytoplasm of the cell, not in the mitochondria. Which means it probably evolved before what? Before eukaryotes. It had to have been before eukaryotes, right? Because they didn't the eukaryotes weren't around at first, supposedly, according to the theory. But remember that during glycolysis, we make, how many ATPs do we actually make? 4. But 2 of them are used as what? What do we have to, we make 4, but we use 2. What are those 2 used for? Activation energy, get it started. So we end up netting 2. Remember, we also make some NADH, which is a source of stored what? And because it's got stored what in it? Electrons. It's electron acceptor, electron carrier. And what's left of the sugar are these 3 carbon molecules called pyruvate or pyruvic acid. Essentially, they're half sugar minus a little bit of energy. At the end of glycolysis, most of the energy that used to be in the sugar is now in which of these things? Nope. Dragon. Most of it's still here. Most of it's still in the pyruvate. Glycolysis doesn't get out a whole lot of energy. Just a little bit. 2 ATPs worth and a little bit of NADH. Computer is not wanting to cooperate very well today. All right, now there's a picture of what NADH looks like. Just from looking at the picture, what kind of chemical is it? Whoops. Yep, it's a nucleic acid. Because anytime we see those rings, what are those? Sugar. And it tells you that's adenine, so that's a what? Charged base, and there's phosphate group. So it's a nucleic acid. Most of those electron carriers are nucleic acids. And this is showing you that NAD plus and NADH are involved in electron accepting or electron giving off. NAD plus gets reduced to become NADH, and NADH gets oxidized to become NAD plus. It's a cycle. It's like, what are we doing? If we're thinking this like a battery, what are we doing to the battery right here? We're charging, and then over here we're using, right? Charging, using, over and over and over and over again. All right, now, like I said before when I was teaching this the first time, you don't need to know all these steps in glycolysis. But just in general, understand what's going on. Right here, we're using ATP in this very first step to do what? Use the word, though. what word are we doing to this glucose?
Phosphorylate.
Phosphorylating it, okay? Which means we're taking the phosphate from the ATP, transferring it over to the glucose. What kind of enzymes do that?
Phosphorylate.
Well, that's a specific one, but just kinases in general, right? Kinases are enzymes that phosphorylate things. What does that do to this glucose 6-phosphate? How does this compare to this? It's a little more unstable, right? So unstable that what happens to it between here and here? It changes shape. That's what do we know about glucose and fructose? They're isomers. Made of the same pieces, but just put together in a little bit different shape. They change shape here. And right here it gets phosphorylated again. And Jake, what enzyme does this second phosphorylation step? Yeah, you know. You just said it. There you go. Phosphofructokinase. That enzyme that Jake likes the name of is an important one because what does it have on it in addition to its normal active site? An allosteric site, feedback site. And what feeds back into that feedback site? ATP. So that when you've done enough cell respiration and you have enough ATP on hand, what happens to this process? It shuts down temporarily. What do we call that? What's negative feedback or feedback inhibition? And it's a way to regulate how fast we need to be doing this. That step is the regulatory step of cell respiration. All right, go a little further here. Now that second phosphorylation step, what happened to the glucose after we phosphorylated it a second time? It split. It broke apart. It was so unstable that it broke. Now we got 3 carbon sugars instead of a 6 carbon. And right here, what is happening to this 3 carbon sugar in that step? It's getting, what's happening to the sugar? It's getting oxidized. Because NAD plus to NADH, that's a reduction thing, right? This is getting reduced. This is getting oxidized. Energy's taken from here and put there. Here we see some ADP becoming ATP. ATP. What's the name for the way we make ATP in glycolysis and kerosene? Substrate level phosphorylation. The energy's taken straight from this substrate and attached to ADP to make ATP. It's different than the way we usually make ATP, which is what? What's the way we make it in electron transport or plants make it in photosynthesis? Chemiosmosis. This is a more primitive way of doing it. Down here we make a little bit more ATP. And we end up with pyruvate or pyruvic acid. 3 carbon sugar minus a few electrons and minus a little what? Energy. And that's where we stop glycolysis. And remember, that happens in the mitochondria, not in the mitochondria, in the cytoplasm of every living cell. Bacteria, plant, animal, fungus, everything in between. Now, if oxygen is available, what's step 2? Well, that's sort of step 1A. What's step 2? Krebs cycle, or citric acid cycle. If oxygen is not available, what's step 2? If there's no oxygen, what's step 2? If there is no oxygen, what's step 2? Fermentation. Okay? Fermentation is step 2 if there's no oxygen. But before we get there, though, let's look at the mitochondria and make sure we know the parts. We know that if you ever get a question about an endosymbiotic hypothesis kind of thing, you ought to talk about the mitochondria, right? What are some things about it that are good evidence of the endosymbiotic hypothesis? It's got 2 membranes. It's got its own DNA that's shaped like a loop, which is the shape of what? Bacterial chromosomes. It kind of looks like a bacteria. It's the same basic size and shape. It's got its own ribosomes that are pretty much just like what? Bacterial ones. And it can do what? Independently of the cell. It can split. It can do like binary fission independently of the cell. So it's a lot like a bacteria. And remember, it's got this inner membrane that's all curvy and twisty. And that's all about what? Surface area. Because what happens on that inner membrane? Electron transport chain, step 3. Remember, the folds in that membrane are called the cristae. And the innermost part of the purple part up here, that's the matrix. What happens in the matrix? Krebs cycle, citric acid cycle. And then we've got this space between the membranes, the inner membrane space. That's important too. We'll get to that in a minute. Don't try to read all this, but remember, between glycolysis and Krebs cycle, we have to take this 3-carbon pyruvate and basically do what to it? Yeah, but what is the pyruvate conversion really all about? We're taking what off of the pyruvate? Pyruvate's a 3-carbon thing. We take a carbon off and oxidize it a little bit. So during the pyruvate conversion, one of the carbons is removed from the pyruvate, and that's what this is saying here, removal of a CO2 from each pyruvate. NAD plus will come in and take away a few electrons too, become reduced. And what's left of that pyruvate, there's 2 carbons that are left. What's the term for a 2-carbon group? Acetyl group. That attaches up with a coenzyme and becomes acetyl coenzyme A. That is the beginning product, the beginning reactant rather, for what? For Krebs cycle. So this happens in between glycolysis and Krebs cycle. It's getting ready to go in. And here we can kind of see the Krebs cycle. Let me see if I've got a better picture of the Krebs cycle. Maybe not. Let's just start with this one. So here we've got pyruvate coming from glycolysis. In this pyruvate conversion step, the CO2 is removed. NAD plus is reduced to become NADH. The 2-carbon group that's left, the acetyl group, attaches to coenzyme A, and here's our 2-carbon acetyl-CoA. That enters into the, where do we say Krebs cycle happens? Matrix of the mitochondria. It goes into the matrix. And it combines with a substance called citrate. I'm going to find a different picture. Let's look at this one. So our this tells us how many carbons we're dealing with. Our 2-carbon acetyl group attaches to this 4-carbon thing called oxaloacetate and makes something called citrate, a 6-carbon thing. That's why the cycle is also called citric acid cycle. And really, the Krebs cycle is all about doing what? What are we doing to this acetyl group? What's happening here and here and here and here? What's it about? Well, try again. Oxidation. Oxidize, 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 oxidize. Which means taking out what? 2 things. Electrons and energy. And we put those electrons and energy in our little electron carriers. Either NAD plus to NADH or FAD to FADH2. So here we do a little oxidation. And when we oxidize, we're taking away electrons. So a lot of times that involves the breaking of what? Bonds. So what starts happening to this 6-carbon substance as we break off bonds? Yeah, but what's happening right here? It's breaking off. It's losing CO2. This 6-carbon thing all of a sudden is a 5-carbon thing. Here we oxidize again, and notice another carbon dioxide breaks off, and now we're down to a 4-carbon thing, and it keeps going. So that's where the CO2 we come from, when we breathe out, comes from. Mostly Krebs cycle. That's where it's made. The CO2 that you're breathing out is the leftover remnants of what you ate, right? You're breathing out what you ate. That's what carbon dioxide is. Here we make a little bit of ATP. And again, we make that ATP by what method? Substrate level phosphorylation. Here we oxidize again. Oxidize again. So the main product of the Krebs cycle is what? NADH and FADH2. Those are the main products of the Krebs cycle. Let me back up one more slide. So by the time we get finished, Between Krebs cycle and the pyruvate conversion together, we make 2 ATPs, 6 CO2s are breathed out. That makes sense because what did we start with? We started with 6 carbons, right? So with glucose. And then we've got 8 NADHs and 2 FADH2s. By the time this step is over, most of the energy that we started with in the sugar, where's it at now? Where's most of the energy that used to be in the sugar at now? It's in these guys. So what's the last step? Electron transport chain, or also called? It's big up. Own it. Oxidative phosphorylation. And that's what we want to look at now. And I'm going to put up a blank slide so we can see that a little bit better here. Oh, I do already have one. Nice. It's called preparation. So, oxidative phosphorylation happens where? In the cristae of the mitochondria, the innermost membrane. So let's draw a mitochondria here. It's kind of an inverted frog, this picture. Alright, so that's supposed to be a mitochondria. What do we call this area right out here? Inner membrane space. And I'm going to draw in. Those are cytochromes. And I'm also going to draw in this guy, which is what? ATP synthase. So remember, here's the matrix. And in here is where Krebs cycle happened. Where NADH mostly got made. The way this is going to work is that this first cytochrome is going to grab on to NADH. And it's going to do what to it? What do those cytochromes do to NADH? Oxidize it. So we're left with some NAD plus and some H plus, both of which are going to be important. But we take away the electrons. Oxidized NADH. And these electrons hop across the ETC from more electronegative one to less electronegative one. And when they're popping, their energy is used to power the what? The pumping of H plus. Now it's a little bit different than it was in photosynthesis. In cell respiration, which way do the H pluses go? They get pumped from in here to out here, right? Get pumped to the inner membrane space. But regardless, this part is still exactly the same as what was happening in photosynthesis. We're trying to build up what? A gradient. It's sort of like charging a battery. Because the more H pluses we have out here, the higher our, what kind of energy? Potential. It's like I said, it's like a battery. You're charging the battery, because those H pluses want to go where? Back in. But they can't, because we've got a phospholipid bilayer there. So we just keep pumping and charging and pumping and charging and pumping and charging. FADH2, the same kind of thing happens too, except what grabs onto it? The second one. So it makes a little bit less, it makes one less ATP, right? But it's the same process. So eventually, our gradient is built up, and this channel here, ATP synthase, opens. And when it opens, what happens? H pluses shoot through really, really fast because of the large what? Gradient. And as they shoot through, we said to think of this enzyme sort of like what? A water wheel. That it can use the energy, it can take a turbine, it can take the kinetic energy of the H pluses and use that to go ADP plus P plus that kinetic energy to make lots of ATP. And again, we call that way of making ATP using this gradient of chemiosmosis. So, let's back up and summarize here. Actually, before we summarize, what have we not talked about yet that we probably should have? Oxygen. Oxygen has not been needed yet, right? Its job is to be the final electron acceptor. Oxygen is famous for being what? Electronegative. So you got these extra electrons sitting here on this last cytochrome. Oxygen grabs onto them, takes them away. And what does that now allow the next electrons back here to do? Come over. But when it takes them away, the oxygen also reacts with what? H pluses make what? Water. And that's where the water comes from in our equation for respiration, right? Our aerobic respiration. So oxygen's job is to take away the electrons from the final cytochrome so that respiration can continue. Without oxygen, it all starts to back up, right? We have to flip over and start doing what? Anaerobic. Otherwise, we run out of ATP and we die, and that's not a good option for most of us. In general, in this step, we make about 32 to 34 ATPs in this step alone, which means we end up with a total of how many for all of respiration?
30.
We get 2 in glycolysis, 2 to the good, 2 net. We get 2 good in Krebs, and we get either 32 or 34 here. So we're somewhere between 36 and 38 total for all of respiration, for all of aerobic respiration. From when what? From one glucose. Now, we got a minute, so let's flip back and let's think about anaerobic for a minute. What kinds of things do anaerobic respiration? Pretty much everything can do anaerobic respiration. Pretty much everything. We only do it to any extent when we're running low on what? Oxygen, typically where? In our muscles. So this would be when you're working out really, really, really hard. You're sprinting. You're maxing out on weights. You're doing something really, really difficult. Just a normal jog or something, probably not enough to make you do too much anaerobic respiration. But there are some things that only do it, like some bacteria, some yeast, and that kind of thing. The first step's glycolysis. Same old glycolysis we talked about before. It makes the same stuff. Again, what gets made during glycolysis? 2 ATPs and 2 pyruvates. Most of the energy that started out in that sugar at the end of glycolysis is again where? The pyruvates. There's no oxygen available, so instead of going Krebs cycle or pyruvate conversion, we flip over and do fermentation. What's the purpose of fermentation? Well, simple terms, it's to keep glycolysis going. Simple terms, is to keep glycolysis going. But here's the deal. To do glycolysis, you've got to have that. You have a limited amount of that. Glycolysis takes that and slowly turns it into NADH, right? So that you're slowly running out of this. That's not a problem if you're doing aerobic respiration, because in an electron transport system, what happens to NADH? It gets oxidized and the arrow goes the other direction. So we're okay. But here we're not doing electron transport chain. So the job of fermentation is to basically take NADH and oxidize it back into NAD+, so that you can continue to do glycolysis. Yeah.
So the NADH that gets oxidized to make NAD+ in the electron transport chain, is that NAD+ taken back and like recycled?
Reused again.
Glycolysis?
It's like a rechargeable battery. Charge it, use it, charge it, use it, charge it, use it. So during fermentation, there's 2 main kinds that we're going to talk about real quick. That's where we'll end today. What kind of fermentation do you do as a mammal, as a vertebrate? Lactic acid. And essentially, you take the pyruvate that you made during glycolysis, the NADH that you also made in glycolysis, You do a little fancy chemistry on those. The purpose for doing fermentation is to make that, NAD+. But as a waste product, we make this chemical called lactic acid. It's chemically related to what? Look at the name. Lactose. It's very similar to lactose in structure, but it's acidic. Which means when you make a lot of it, you what? You feel it, you burn. And this causes muscle what? Fatigue, soreness. And that's why if you really work out, especially if you're in bad shape, the next day you know it, okay? Now the reason cheaters like Lance Armstrong were so good at what they were doing is they cheated so that they didn't have to what? They didn't have to do anaerobic respiration, which meant they didn't get muscle fatigue and they didn't get muscle soreness. So they could just keep on paddling in robotics.
Attention Joy Club members, there's a meeting in the Clements room right now.
Now that's lactic acid fermentation. The other kind of fermentation you need to know about is what? Alcoholic. Essentially, same purpose. Take the pyruvate, use it to oxidize the NAD plus back to NADH. But instead of giving out lactic acid, what are the two waste products of alcoholic fermentation? CO2. That's why beer has bubbles, right? And ethanol. Ethyl alcohol, ethanol.
The same stuff that goes in our cars.
Yeah. If it can run your car, it's probably not good to drink. Now, people who drink a lot get fat. Why?
Because they have a sedentary.
Well, yeah, that's part of it, but related to that, why?
Energy, what energy?
That's got almost all the energy that originally started where? In the sugar, in the glucose. Because how much energy total do we get out of the glucose if we're only doing this? If we're only doing anaerobic, how much energy do we get out? Just 2 ATPs. So there's still about how many ATPs worth of energy left in that ethanol?
34.
34, maybe 36. There's a lot of energy left. So if you take in a lot of ethanol, you're taking in a lot of calories. And beer or wine or whatever doesn't just have ethanol. What else has it got? It's got sugar. So that's extra calories on top of that. It's calorie heavy. And it's going to make you gain some weight if you take in a lot of it. Now, one other thing I want to talk about, and then I will zip it and let you go. And this is kind of an important thing. If you don't eat sugar, let's say you get on one of those protein diets, those, what they call them caveman diets right now, right? It's kind of a fad right now. You know what I'm talking about? Caveman diet?
Yeah.
You're eating mostly meat. Can you do glycolysis very well if you're eating only meat? Not so much, because you need to start with sugar, right? But you still need to get some energy. You need to burn that protein. Your body's first going to digest the proteins and break it up into what? Amino acids. Now here's an amino acid. What do we call this group over here? The carboxyl. And this is amino. And then we've got this R group down here. Now here's the deal. Let's say that that's the only thing that we're eating, or maybe we need to burn it to get some energy. The first thing your body's going to do is it's going to take off this amino group. It's going to break that bond right there. And that amino group reacts with what? A hydrogen. What did we just make? Ammonia. Is that a good thing? Ammonia's pretty toxic stuff, right? It's what's in like Mr. Clean and Windex and cleaners. One of the main jobs of your urinary system is to get rid of that stuff, right? And we'll talk about that. But once we take off that amino group, what do we essentially have right here? What is that? 1, 2. What is it? It's an acetyl group. So that's almost like we already did what? It's like we already did glycolysis and already did the pyruvate conversion. So now that what's left of that piece of the amino acid can go straight into what? Well, not electron transport. Straight into Krebs cycle, and then it can go to electron transport, and we can get energy from it. Sort of skips glycolysis, skips the pyruvate conversion, and goes straight into Krebs cycle to the citric acid. Does that make sense? But the downside of that is you're generating all this ammonia. And your kidneys, and it turns out your liver also have to work on that because ammonia will kill you if you don't have a way to deal with it. We'll talk about this a little bit more next week. Your body, in mammals, and also in amphibians, our bodies are going to take this ammonia and convert it into something a little bit less toxic called urea. And that's what you end up urinating out. And we'll pick up with that. Okay. That's it. And I need to get the sign-in sheet from whoever's got